apologies about the slow start. We had a few things against us. We had yoga after that, and we couldn't very well move them on. They had the room booked till 1. So thank you for your patience. My name's Judy Blackbeard, and I manage the applied research team. Nick is in that team, and I assume you all know Nick, seeing he's sent the invitation out. So what I want to do is just to welcome you to the launch of the Nuisance and Harmful Algae Science Practice Partnership. Today represents the culmination of a lot of hard work. Establishing a research partnership requires vision, it requires hard work, and it requires compromise. So well done to everybody who's been involved in getting this up and going. And at Melbourne Water, we're looking forward to the research and the outcomes that are going to result in this partnership. I also want to thank our interstate visitors, William and Fiona, thank you very much, and Greg came from Newcastle, so possibly a little bit longer. And I was just hoping nobody had to get up before 6 o'clock, because it really is killing when it gets to this time if you've been up since 4 to get an interstate flight. And thank you too to those locals who have come and those not so locals who've come. I'm really delighted that you're interested, and we hope you find this interesting and the research also proves of benefit to you. Just very briefly, safety, we can't ignore that. A lot of you came up the stairs. If you go that way and turn left, you'll see the staircases that would go straight down. So if we have to get out in a hurry, that would be the way to get down. Don't use the lifts. And if you need the toilets, the lifts are in that direction over there, ladies on the far side and gents on the closer side. So now I want to hand over to Nick, who's going to introduce the partnership.
Thanks very much, Judy. As you can see, it's a pretty compressed agenda, so we're moving quickly through these presentations, and please save your questions until the end. Otherwise, our GM will never get to launch. So I'll just give you a very brief introduction as to why we decided to invest in this research partnership. I'm not very good at acronyms, so I'm pronouncing it as NASP, Nuisance and Harmful Algae Science Practice Partnership, which has a vision, of course, great science and engineering to support our theme at Melbourne Water of life and livability. The last bit's obviously important because algal growth is very much enhanced through increased temperatures, particularly cyanobacteria. And this is really a mix between science and engineering, this partnership. The partners are the New South Wales Global Water Institute, the Water and Algae Hall, Institute of Medical Research, and Canadian University. That last bit came about because a key person in the partnership has taken up residence back in his old digs. So we're continuing that collaboration through him. So the themes that the partnership cover are pretty much everything to do with cyanobacteria in the water industry. So prediction and asset design, risk and surveillance, treatment and prevention, and then translation of that information through to industry. We're covering all of these themes because at Melbourne Water are a little bit unusual in that we have water, sewer, and also waterways. So cyanobacteria in particular impact on all of those product streams in different ways. For example, it's pretty hard to treat a river, but that might be a solution in the reservoir. So surveillance becomes perhaps a little bit more critical in some contexts versus others. The projects are in their formational phase, but we have outlined a series of them, and I've tried here to assign them to different types of projects, operational, strategic, and roughly what the time period is. So why has Melbourne Water initiated this partnership? Well, we have at the moment, every year we have toxic algal blooms at Western Treatment Plant, which is the world's largest lagoon-based wastewater facility. And I'll talk a bit about that later on. We have taste and odour issues at 2 drinking water reservoirs. And we're also the regional BGA coordinators, so we deal with a lot of shallow water bodies which are bloom producing, and that chews up quite a bit of internal resource. So just on that, if you look at, this is from the department's BGA portal, data between 2004 and 2016, and these are blooms reported to the portal, so it's exceeding some trigger level, and you can see there that clearly the big bloom areas are reservoirs and urban lakes. We've got a lot of urban lakes, some of them not that well designed. The SDP bit is probably a bit underplayed in this graph because Western Treatment Plant doesn't fit into this data set. These are the bloomers in our area. So Dolichos sperm used to be Anabina, so essentially it's Microcystis and Anabina and assorted others are the critters that contribute to blooms that are reported through to the portal. But we are seeing some unusual blooms as well. This was a bloom just here. To the left is a very small coccoid cyanobacteria called Radiocystis, which bloomed in our Greenvale Reservoir this year. And it's been artificially mixed by ALS with Microcystis just to show the difference between Radiocystis and Microcystis. And as you can see, it's very small, and these things are really hard to identify down a microscope, which is why it's helpful to have molecular and other techniques which can complement microscopic data. Oh, yes, and all the literature was from Brazil, so this was a bit unexpected to have that blooming in our reservoir. And a couple of later speakers did some work on that for us to confirm that it didn't have the capacity to produce toxins. So that's Greenvale Reservoir. And then just a few weeks ago, we had the first nodularia bloom in the Melbourne region, which occurred in a large pond that is situated near a bird high. And it seems that this pond interacts with the groundwater because the EC is quite high. Nodularia likes high EC conditions, brackish water. It was confirmed as carrying the nodularia gene, right? Yep. So potentially toxic bloom. Here's Western Treatment Plant. As I said, the world's largest lagoon-based wastewater system. Just to give you an idea of scale for those who aren't local, that there is many tens of hectares, just that one system. It's easily visible in Google Maps, for example. And it has a history of Microcystis blooms. We had a very large bloom this year in this pond here. If you Google Louis Helbig, who's an artist, and flew over Western Treatment Plant during the bloom, you'll see a very spectacular picture of a Microcystis bloom in that pond. But what does that mean for us? Well, we produce Class A and Class C water, and when we have exceedances above a certain threshold, then we can't deliver that water. And through the period 2009 to 2016, there are roughly 173 days where we need to notify our customers with respect to Class A water. So cyanobacteria Western Treatment Plant does cause us issues. Fewer notification days for Class C water. Thresholds are a lot higher there for notification. And the entire Western Treatment Plant site is a Ramsar site. So one of my concerns is that around the world there have been quite a number of mass casualties of birds, some in the tens of thousands, related to toxic algal blooms. So this is a risk at Western Treatment Plant. And at some time, particularly in the middle of a drought, we can have most of one species of duck in Victoria sitting on Lake Borry at Western Treatment Plant. Here is, I guess, the overall reasons why we thought that the strategic approach was the way to go. So this is known everywhere, nothing new here. Global warming appears to be driving conditions that would favour particularly cyanobacteria. They love warm, still water bodies. In cities which are aggressively urbanising, like Melbourne, we will find more Australians and wildlife living in in closer proximity to shallow water bodies. We are still finding new toxins, new taste and odour compounds, and in new taxa and in new environments. And water authorities are resource stretched, so we want methods which are quick, reliable and cheap. Here are the known genera, now at 37, that produce toxins, and this is still being added to. And I just thought I'd finish with this one. So, global change is certainly contributing to big changes around the world, and atmospheric transport of microorganisms is something we know very little about. But we do know that in Victoria, the expansion of the Hadley cell is driving quite a bit of our change in climate. There's a review recently by the department around what is changing Victoria's climate, and expanding Hadley cell is a key part of that. And so we should expect there to be changes just through that in the distribution of microorganisms as they circulate through these Hadley cells. Here is a bloom recently, 2015, in Murray River, and this was the first bloom of Chrysospora. And it was a very large bloom that had an impact on the tourism there. So we're seeing around the world an expansion of certain taxa, and also new taxa occurring where they didn't before. Of course, it's difficult to know whether that's increased surveillance or not. It is complicated, but there's a reasonable amount of published and unpublished information which suggests that this is occurring. And I'll hand over to William.
Thanks, Nick. So Greg Leslie from the University of New South Wales, who's the director of the University of New South Wales Global Water Institute, was supposed to do a brief introduction, but he's unfortunately ill and unable to make it today. So William, also from UNSW, has very kindly agreed to step in. So I'll just quickly run through his bio, then I don't need to do it later when you do your own talk. So William's an associate professor and principal research fellow at the University of New South Wales Water Research Laboratory in the School of Civil and Environmental Engineering. His primary fields of interest are related to estuarine hydrodynamics and water quality, including restoration of estuarine environments, acid sulfate soils, coastal wetlands, boat wake waves, that sounds quite fun, outfall hydraulics, and field testing and related physical and numerical models. So thank you, William. If you can.
Thanks for having me. I never know what I'm putting those things, so I appreciate it. Oh yes, I'm stepping in for Greg. Greg is the director. It's a really interesting organization. Am I going the wrong way? And so we have this thing called the Global Water Institute, and it's bigger than Ben-Hur. It's a huge thing, and I've been fortunate enough to travel around the world with Greg and the director before Greg, spruiking the benefits of the Global Water Institute. So what is it, and what do we do? What we found at the University of New South Wales is there are many, many academics in different faculties, in different areas that all somehow work with water, but weren't working together and weren't using our knowledge cumulatively to actually do something bigger than the individual. So they got together this idea that if we had a global water institute, we'd be able to sort of have a focus and a drive and go global. And that's been the aim of what we call the GWI. So there's 400 of us. We include professional academic PhDs from 7 faculties and 13 research centers just within the one university. And this is why we thought we'd better start working together. So we exist to provide excellence in water education, to try to connect that area between, I guess, the applied and the fundamental. That's a big issue right now for us. And to make sure that people that want to work in that space know that we're there to work with you. So I think Greg would be remiss if I didn't say this is an ideal situation for us, where there's a utility that is particularly interested in something, and they can grab people not just from one area of the science, but from all over. So I'm at Manlyvale out in Water Research Laboratory. Fiona is in the main campus, and then the other people involved are in even different schools and in different buildings. So we don't normally work together on these things until they brought us all together and said, if you focus this attention, it might be of value. And we work in a whole range of areas, so there's everything from water to water resources to coastal and aquatics, and I'll talk a bit about what I do, and Fiona will talk about what she does. There are other people involved in this group that aren't here that would send their apologies, but they do a lot more in lines of the actual algal growth and I guess the algal risk. And I've been fortunate to take this message, and we've gone to China together, and we've gone all around Australia, and they've been to Vietnam and other places selling the idea that if you need people in water, we've got hundreds. And that's probably all I have to say.
That was very short and sweet. We'll be back on time if we're not careful. So our next speaker is Brett Nealon. Professor Brett Nealon is head of the School of Environmental and Life Sciences at the University of Newcastle. He's an expert in molecular microbiology, genetic and genomic engineering, and microbial chemistry, and has published extensively on the genetics and biosynthesis of toxins produced by cyanobacteria. So welcome, and we look forward to what you've got to say.
So this, I want to go through, I think a really interesting project that's been neglected for a long time in cyanobacterial research in Australia. And Australia is one of the countries that really started a lot of cyanobacterial research way back in the early 90s when we had a big problem in the Darling-Barwon river with toxic anabina producing saxitoxin. What has been totally neglected is the input of the benthos as a seeding environment for the blooms that we see on the surface. So we formulated this project, and it's about to be submitted as a linkage grant, hopefully in the next 2 weeks. We've been working on it for about a year. And the main players are Aaron, who's here, Nick, and Caitlin, who is a Water Research Australia PhD student. So we're going to focus on Microcystis aeruginosa because in the Western treatment plant ponds, we figure this is the main nuisance or harmful algae that we're going to be targeting. But as Nick said, there's plenty of other species present. The reason I'm part of this group, I guess, is because historically we've been studying sorry. What's your language? It's all right. I mean, I'm not at uni anymore. Yeah, that's right. Is there a pointer? Now lecturer steps in, right? This is microcystin. It's a cyclic peptide. We discovered the genes that create this molecule. So essentially, I'm coming from the point of view of being a geneticist, a molecular biologist, understanding why these toxic compounds are made. The importance of this project, I think, is understanding why they make the toxins, and are these toxins also present in the benthos, as we know they are in the planktic environment. All cyanobacteria have these wonderful physiologies that they've developed over 3 billion years on Earth, and issues about phosphate and nitrogen metabolism, their dormancy, which is really the focus of this project, how they survive over winter and all of a sudden pop up in the summer. It's not creationism, that they have to come from somewhere. So the physiology in this benthos is really something that's never been studied before globally. So this really puts our project here with Melbourne Water on the historical map. So there's many different toxins that can be produced by different species, and there's different isoforms of those toxins. We want to understand what selects for the different toxins, the different isoforms, because they all have different toxicities to humans and birds. So what happens is we might study it here in WTP, but there's all different issues related to what species are selected and then what toxin in those species is being produced. As you'll see in our long-term plan, we want to overlay the data that we generate from W2P across many different water environments in Australia and the world. So the crux of this project is that this is typically what's in winter, or we're predicting what's happening in winter. And WTP, I should say, is a classic environment for this study because the only input is sewage. Am I correct? There's, like when we're talking, we've done lots of work in rivers, and this question is very hard to answer in rivers because you're going to have input of toxic cyanobacteria flowing in from tributaries into a river and then into a reservoir. We've done lots of work there, but we can't answer the question in a reservoir where the toxic cyanobacteria come from. It's either coming in from the tributaries or it's actually in the benthos, deep in the benthos of these reservoirs, which a lot of people would say couldn't be possible because they're too deep at the dam wall, for instance. So again, WTP is a classic model for this type of study. So this is the overall aim or the overall goal of our linkage project that we've formulated. So we want to study the biota, so the algae, the cyanobacteria, and the metadata or the abiotic factors that go along with what happens when a bloom is formed or not, and whether that bloom is toxic or not. As I said, we want to understand dormancy, and this plays with the climate change issue that Fiona will talk about later. Is with climate change, will we see less dormancy? Will we see less of this, what we call wintering event? The stability of the population that's present, because we're using genetic tools here, we call it genetic stability, and what conditions, what environmental conditions lead to that becoming a planktic bloom. So Nick's already shown you this. This is the site we're using. Correct me if I'm wrong, Nick, but this is is it Eastern?
25 Western.
Western? I'm going to get some.
55 East.
And the main studies in
It's one of
that
the
area.
sites, yeah.
So we've done some initial work, and I just want to point out here, when we look at Microcystis in the purple line, it is the most variable population that we see. And this is happening not just over winters, but even in the summer months. So we get this variability of the population present. I'll go back to an example in Warragamba Dam in Sydney, where we did a similar study using quantitative PCR to look at the genotypes present in the water. And whereas you might get a whole population of Microcystis, at a certain time of year, you'll get a spike in a toxic subspecies, a strain. And this is something you won't pick up with microscopy because they all look the same under the microscope. But by using genetics, we picked up that the bloom in these November months were not toxic and therefore didn't need extra filtration treatment by the water managers, but all of a sudden you get a spike in a toxic population and hence the water becoming not potable. So the first aim, we have three aims in this project. The first aim is to characterise the population that's already in the WTP. And again, Nick's has some data and that's microscopic evidence. But obviously there's other things there that we need to characterise. So we're going to do genotyping of the benthic population and the planktic population. We'll do toxin analysis, and that's chemical. And we'll use methods with Aaron here at Weehigh. We'll do a metagenomic analysis, and that'll tell us basically the whole bacterial or algal population that's present in those two environments, the planktic and the benthic. We'll then understand the community structure over different times of the year, actually over 2 years. So you can see under different conditions of nitrogen and phosphorus, you do get different types of strains being present, and these different strains also have different levels of toxicity. For example, if you look at microcystin production, which is the toxin from Microcystis, high nitrogen, high phosphorus and low iron all lead to increased production of toxin. And that could either be that the genes are switching on, their toxin genes, or different species are being present, so the genetic stability of the population could change based on the available nutrients. Aim 2 is to then look at the genetic regulation. So the population is the genotype, and now we're looking at the phenotype. So what genes are expressed, and therefore we're going to use transcriptomics, which is looking at the RNA instead of the DNA, and the proteomes, which are looking at the enzymes that produce the toxins. So this is more the functional analysis of what's happening in the water and the benthos. For example, the transcription or the RNA increases in highlight or red light, which you get. So this is another depth issue. So the W2P is a shallow water body that I think Will will talk about a bit later as well. And the starvation of iron. These factors will feed into AIM-3, which are what model do we use to predict blooms and predict the toxicity of blooms. So that's the abiotic factors, the levels of nutrients present. This will all feed into a predictive computational model, which will be mostly done here in Melbourne Water at the latter stages of the project. Aaron produced this wonderful diagram that's going in the grant. It's confusing to me, but you understand what it is. And the reviewers of the grant are obviously more intelligent than I am anyway. So last two slides, we've formulated this project with short-term and long-term goals. So in the short term, we want to understand what is there and how can we get genetic markers to tell us or to monitor that water over time, over monthly sampling periods. We use the genomics approach to find out what is the difference between what's in the benthos and what's in the plankton population. In the midterm, we will develop a model that's applicable to WTP, and in the long term, we want to actually extend that model to places, to rivers, which I said are incredibly difficult to monitor right now, but with the model of WTP to start with, we can maybe extend it to rivers and reservoirs. That's it.
Thank you very much. Now our next speaker is going to be Aaron Jacks. So Aaron's an associate professor he's the laboratory head in the Division of Population Health and Immunity at the Walter and Eliza Hall Institute of Medical Research, and a senior lecturer in parasitology in the Faculty of Veterinary and Agricultural Science at Melbourne Uni. Aaron's had a long history of working with the Victorian water industry. He's worked with us for many years, and he's helped to develop tools to monitor for aquatic microorganisms that present a public health risk. So thank you, Aaron.
Thank you very much. So continuing on from the work that Brett was discussing, and as Judy said, I was going to give us a little snapshot of some of the work we've been doing with blue-green algae over the last few years, basically around developing improved tools for the diagnosis and environmental samples. So as Brett already introduced, there are obviously, and we've talked about already, there are a number of toxic cyanobacteria that exist in the environment, and within the Australian context, historically, these have included those organisms that can produce microcystins, nodularians, saxitoxins, and cylindrospermopsins. As Brett and as Nick have already said, you cannot differentiate toxic from non-toxic varieties of these different taxa based on microscopy data alone. So we wanted to be able to develop tools that will allow us to somehow tell which variants are toxic and which aren't in terms of understanding the potential health impacts of a bloom in any given circumstance. As Brett said, Brett's group in particular has led research on understanding the biochemistry, the molecular biology that underpins the production of cyanotoxins, and has identified, he in principle and other researchers globally, have identified a number of these toxic gene operants. Brett talked about the one for microcystin, but ones have been identified for each of the cyanotoxin producers. And we can then target those genes that are intrinsic to the makeup of that synthetic cluster that are essential for the production of a cyanotoxin of any given type by a method called the polymerase chain reactions, a DNA-based method, probably many of you are familiar with, where you can amplify a targeted region of DNA that's diagnostic for, in this case, one of the genes that's essential to make a cyanotoxin of interest. We can tell whether it's a microcystin producer or a cylindrospermopsin producer, and so on. So that led to a project that Judy Blackwood in particular was really instrumental in getting up off the ground, which was funded by Water Research Australia and of course Melbourne Water and other partners in Victoria in the water industry, which was tasked with trying to develop a rapid molecular diagnostic tool that we could use for routine surveillance of toxic cyanobacteria, in particular the four major toxin producers in Australia that I mentioned. We partnered with a Sydney-based company called Oz Diagnostics to develop a technique, well not develop a technique, but to apply a technique that they developed called multiplex tandem PCR, where you have a series of PCR primers that amplify on bulk a particular complex or multiplex of genes, in this case each essential, one of 4 essential genes required for toxin production for each of the major toxin classes, and then a specific internal primer set which allows you to quantify how much of that toxin gene is present. So you can basically estimate the approximate contribution of each toxin producer in your initial, just in case of a water sample. And this work was published in Molecular Cellular Probes, a scientific journal that specializes in molecular diagnostic tools, in 2013 with Louise Baker as the postdoc in my lab who led that project. And by comparing the data that we got from here with conventional PCR-based tools, sequencing to determine that what we amplified was indeed the gene that we were interested in for each of the toxins, and also environmental-based detection of toxins using the Abraxas dipstick, ELISA-based dipsticks that directly detect the toxin, we were able to show that our assay had 100% diagnostic specificity, meaning we were not getting any false positives. Every time we saw a positive, it indeed was what we were looking for, and 97% sensitivity, which means we had 1 or 2 false negatives. Those false negatives were identified based on, these were a couple of saxitoxin producers, where we had trace levels of the saxitoxin toxin, but we couldn't identify the presence of the gene in the sample. Most likely the explanation for that is that toxin is hung around in the environment, the balloon has crashed, we can no longer detect the DNA. And so from that perspective, I don't think these are true false negatives, but nonetheless, that was what our data told us. Moving forward from then, in 2014, Nijoy John, who's here in the audience, joined my lab as a PhD student, again funded through Melbourne Water and also through Victoria Water. And his project was looking at basically validating the use of this cyanotoxin test in the field, and also developing some additional applications around these approaches. One of the areas that Nijoy's done a lot of contribution has been in testing this large bloom that Nick mentioned and applying our MT-PCR technique to try and understand what the presence of toxins in these samples were, and what that told us about the complexity of the bloom. Within the context of that Murray River bloom, we had a lot of microscopy data, which, as Nick already said, showed the predominant species was Chrysosporum mobilosporum, but there were other species present. We could see some that had the potential, such as Cylindrospermopsis, to produce microcystis, or to produce in that case Cylindrospermopsin, and microcystins that could produce microcystis, and so on. We then tested that using this MT-PCR technique, and Nijoy showed a complex mixture of low levels but present toxin gene based on gene copy toxin producers that could make each of these 3 saxitoxin, Cylindrospermopsin, and microcystin toxins. We showed that there were probably 2 different bloom complexes, that one perhaps successionally following the other, or possibly arising from 2 independent starting points, that were dominated in the first case throughout much of the bloom with this Cylindrospermopsin producers, and further down the river with microcystin producers. There was one little spot here near Yarrawonga where you also saw some microcystin producers predominating. We then confirmed that by sequencing, so this is just taking that gene sequence and identifying that indeed these are microcystin producers or Cylindrospermopsin producers, and there was also low-level positive results for both microcystin and Cylindrospermopsin toxins based on the ELISA-based detection methods, and also using a technique called high-pressure liquid chromatography. What was interesting about this is in this area of the bloom we saw, or the river, we saw very relatively high levels of microcystin production, but on microscopy, nothing that we could point to that indicated a known microcystin producer. We're not sure where the source of those microcystins are. We can confirm the gene is present. Nijoy's also moved on, sorry, this is just the data on the conventional PCR, I'll move past that. Nijoy's also moved on, this is, sorry, just to touch, this is just showing that we were able to, using conventional primers, also amplify microcystin and Cylindrospermopsin producers. So they were true positives, that's all I was trying to show in that slide. Moving on, Nijoy also started to look at trying to improve our capacity to do molecular diagnostics on other types of nuisance cyanobacteria, in particular cyanobacteria that produce taste and odor compounds such as geosmin and 2-mid. Again, lots of different organisms can produce these compounds, but being able to predict them using a molecular tool would be very helpful. Each of these, as with cyanotoxins, the genes that are responsible for synthesis of these compounds are known. In this case, just a simple gene, a synthetase gene called geosin synthetase. We went about, or Nijoy went about targeting in Victoria, blooms that produced these, or that had the presence or absence of this gene to try and develop an improved diagnostic tool. There are PCR-based tools to allow geosmin prediction or geosmin detection. Unfortunately, geosmin is a very variable gene, and there are a number of different protocols that have been developed for its amplification by PCR. And each of them requires a separate set of PCR primers and a separate protocol, and none of them will pick up all geosmin producers. In effect, what that means is that if you want to screen routinely for geosmin-producing cyanobacteria using this technique, you need to run 3 separate protocols. You've got a lot of triple handling of samples, it's increased cost, increased time, and so on. What we wanted to do was see if we could understand more about the genetics of this gene in blooms in a number of different locations. Could we identify, at least in Victoria and then possibly expand further, a single primer set that we could use as a universal test that would allow routine monitoring? So that's what NIJOI did. Again, as I said, you know the genes that are responsible for producing these. NIJOI then used the existing 3 primer sets that had previously been published to amplify the full-length genes, to sequence them from about 250 samples. This included all of the samples that were collected over the original Project 1043 that I mentioned that led to the production of the initial cyanidist that we produced with Water Research Australia, as well as samples collected over 3 years of the course of NIJOI's thesis, including all the samples in the Murray River bloom, as well as samples throughout Victoria that were provided to us through Melbourne Water and through Victoria Water and so on. He got 85 samples within these 253 that were positive for geosmin based on one of the 3 PCR primer sets, and there was no single published PCR primer set that could detect all of these. He then sequenced them and then took a sequence analysis to try and identify what was present. One of the things we saw is that this Dolichospermum, this works. That Dolichospermum eucranicum was by far and away the predominant geosmin producer across all locations. We then undertook, or NIJOI then undertook, what's called a gene clustering analysis. This is a phylogenetic tree. It doesn't matter how this works, but basically everything that clusters together is very similar from a genetic perspective. These are all the different clone types that NIJOI identified within these Victorian samples, as well as several known Dolichospermum eucranica and related species. This large cluster and a few other little ones. NIJOI then undertook something called a sliding window analysis, where you're basically looking across the length of the gene. And what you're looking at here is the higher up this blue line goes on the chart, the more variability there is in the gene. NIJOI located a region also shown in this graph here, where you have very low levels of genetic variability across a number of different taxa. All this little square chart shows is these are different species of, or different gene sequences of geosmin producing cyanobacteria, and where you've got a little nice blue spot is where there's very little genetic variation, which is where we'd like to target a primer. Did that and then tested those primers against all of the samples that we previously studied, and NIJOI showed you could amplify every last sample that we had previously detected with one of the 3 using just this one primer set. So you have a single user-friendly and universal protocol for at least all the geosmin producers that we're interested in. Last slide in terms of future work. NIJOI is in the process now of putting that geosmin assay, as well as an assay targeting antitoxin-producing cyanobacteria, onto and adding that onto the MT-PCR assay that we created. He's also done some conventional testing for antitoxin producers in the Murray bloom, and has found actually quite a few that have this antitoxin-producing gene, which is interesting because that previously hadn't been thought to be present in Australia. And then we're looking at a few other techniques to try and do point of source detection of cytotoxins. I'll conclude there and thank many, many people that have been involved in this project, and thank you for your attention.
I wasn't aware you'd cracked that one. Well done.
And a new toxin.
And any talks?
Yes.
Well done.
Look, our next speaker is William Gilmore. So I've introduced you already. I don't have to introduce you again. Rick will get that up, and welcome.
It's the second one there from the top. That one in. So now for something completely different. This is as far different as you can imagine. So, so the process that we've gone through is, again, we're talking, and I'm talking about GWI, is that we work in all different spheres here, and I work about as far away from that sphere that we just talked about as you might imagine. So I work on the big scale and catchment-wide scale of how we do things. So my take has largely been, this guy there sitting at the swamp going, this place looks really bad, what do we do? And I take that from a hydrologist, environmental engineer, where each world has equal importance. So I'm an engineer who works in the environment, and what we're trying to do is take sites from this to that, and whatever that may be, whatever the problem is, we try to turn them through re-engineering things back to something with a bit of design, a bit of process to get a favorable outcome. So I thought if I've got an audience, I should tell you that I do a few things, and it's been a very busy week. I work quite closely in the PFAS space. I chair the Williamstown PFAS work for everything related to water, which has been a very big week with Four Corners and everything happening there. I do a lot of work with Hunter Water on estuarine dynamics, so we've got a big study up on how they might look at their entire estuary, understand everything that's happening in their estuary from the microbial loop right up to the fish and birds, and then actually work out what their licensing structure might be for their outfalls. So do they want individual licenses, or they want bubble licensing for the whole thing? And how we can do that is if you know how the whole river works. That project's underway. I did mention boatways. I do a lot of work in understanding the impact of boats and rivers. I'm chair of PA Australia, which is a big thing working in the maritime industry. And then we do a lot of work in point source. So how do you discharge things when you go back into the environment? Where I work is in Manly Vale. It's the Water Research Laboratory. We have many, many large sheds. With the volume of water fed from Manly Dam, we can do about 700 liters a second, and we pump stuff through big physical scale models. 700 liters a second is loud, and it's a lot of water moving around at a lot of times. So we do scale modeling of how you might put things out and make sure that they have no impact wherever they're going. So we design most of the desalination outfalls in Australia, and this is a picture of the Clarence River. It's the biggest river in New South Wales, and we designed 2 ebb tide releases here that only discharge on the ebb tide and make sure that everything that's discharged clears the entrance before it comes back, and a whole range of problems in that space. But today I'm just going to talk about this water body design and restoration. So my general feel for why we are in this predicament we're in is that we've taken a very nice environment and we've completely carved it up as much as possible. So we've taken these environments and straightened them and channelized them and drained them, floodgated them, cleared them, and done just about everything else you can imagine to the sides. And then we wonder why we have a problem at the end and why water quality isn't as perfect as it should be. And we have things, so this is some historical pictures of this is the Richmond River catchment in New South Wales that went from this to that. Every red line is a drain that's gone through it, and it's taken out all of those little water bodies that were there before. And I have a whole presentation where I just show that all the sites around the country have gone from these beautiful sites to just a whole number of drains and paddocks where residence times have changed and the amount of water quality that it can actually produce has changed. And it's happened everywhere, and it happened in a really short time, and most of the people have forgotten that it even happened before. So when they look at the estuary, they think the floodplain is something quite distinct from it. When in reality, the floodplain is the estuary. We just put a levee between the two of them, and that's why we've lost a lot of our water quality. And for example, this is what most of the sites look like. So there used to be no drain through the middle of this, and a large water body that filtered the water, filled with trees, and a natural flow of hydraulic residence time. Now there's big drains, and every one of those red lines was a drain like that. And it was done systematically, with a large amount of money, with lots of people working through very unique methods, from drag lines, so this is dynamiting out channels to clear waterways, to a whole range of scary looking things that dug up the landscape and changed it. And that's led to serious implications. So we have water quality issues across the country in many, many places that were predicted by a few beforehand and have now come to fruition in many, many cases. And slowly we're trying to find few spots and turn back the tide. So this is an article from 1977 about oyster fish kills, and this is the same exact side, an article that I was involved in 2017 saying same thing's happening and we've done very little to fix it. And that's not just individual sites, that's whole ecosystems. So we've had a serious impact on fish habitat, we've had an impact on bird habitat. Largely most of our large floodplains are now, I think, Nick mentioned before, there's SDP, where the treatment plant is the largest area for most of the birds. Well, that certainly didn't exist previously. And we got to the point, at least in New South Wales, where most of these systems, the ecosystem of cells are endangered. So all of our salt marsh is endangered, and we've lost most of it. So my work has been, how do we fix that? How do we physically reverse some of that, and how do we find out what is the best way of doing it? I'm not saying we're going to do it everywhere, but there are priority spots where we can get in that what we're seeing is 80% of the problem comes from 20% of the landscape. And if we can get in and actually fix up that landscape, we make a huge difference to everything else. So we're taking this landscape and just bringing a little bit back, but it's the key bits that matter. So I'm an engineer working the environment, so I do the physical dynamics. We have a group of people, we're all doing the physics, and Ken will talk more from the physics perspective as well. We then work with all the ecologists who know all these things that are important about how the bugs work, how the birds fly, where the fish want to eat, and we create these solutions that then get rolled out on site. My fundamental thought is that underneath all this process is getting the water right. If you don't get the water balance right, if you don't get the retentions timed right, the flushing times right, if we don't work out the mass balance well, we're never going to work our way up through this chain to get those pretty birds you want in the picture at the end. And very often we find when the system doesn't work, it's because they got it wrong to begin with. So what do? We start by prioritizing the landscape. So we're looking at places where there's problems. It could be algal problems, or it could be acid-soluble soils, or it could be the destruction of some habitat. And then we work out what is the best bang for a buck in these landscapes. So we prioritize where are the biggest hits. We do this traditionally across the whole catchment. What is the biggest problem for the area? And then we come up with priority plans for each individual area, saying this is the highest land, the biggest problem you have in that whole area is this one little bit here. If you got that little bit fixed up, we'd solve 80% of the problem. And that's what we're finding across the area, is that most of the problems come from a part of the outcome. Once we've done the priority work, we spend a long time doing the dynamics. So working out how the system operates, either that could be incorporating the microbiological dynamics or whatever it may be. So this is geochemical dynamics, and this is hydrodynamics, looking at how the whole estuarine function changes as the water flushes in and out of the system, and what that does to the water quality, and particularly in this case the algal response. So once we understand how the system works, then we might be able to go and do something. So where we do a lot of our research space is in the what to actually do. So it's that design component. So again, the engineering side, that we have a whole range of options that we might apply on site. So making the site wider and shallower, putting benches in, or redesigning the fetch, or looking at the landscape in total, restoring it in some way or changing it in some way, and then we physically go do it. So a bunch of before and after pictures. Here's what a site looked like. Here's what it looks like now. Same exact location. This is in Newcastle before and after. This is a large site on the Hawkesbury that we've done a lot of work in the algal space. Before and after, trying to generate a landscape, it's about 35 hectares, where we're generating salt marsh for increasing bird habitat. This is an area where we're looking at using greenways to encourage wetland design. So redesigning the landscape so we can use greenways from your actual bins to recreate the landscape. Big site, 1,000 hectares here for before and after, where we've completely restored the landscape. This is a 3,500 hectare site with a process of redesigning the whole landscape and changing it back into where we're going to put the water and how we're going to hold it. This is everlasting swamp, again about 4,000 hectares before and after. We're looking at large scale restoration. The big pictures are more fun, but we work on the small scale too. And big swamp, which is the cover for what not to do, cover story for what not to do. Before and after, it's about 2,500 hectares. Before and after, completely scalded, nothing living. After, before and after. So we filled in 28 kilometers of drains. We redesigned a river that's 2 kilometers long, 500 meters wide, and we're redesigning everything with the idea that the fundamental microbial cycles are worked all the way through to the end game, and going to work out with the right process. So there's a whole lot of engineering that goes in to get you to that end process. So we're bringing the hydrology and all the downstream considerations and upstream. It's not as simple as just fixing the actual pond. We have to integrate all the eco-hydraulics and look at the values and, of course, on-site management, how you're physically going to manage the site into the future and who's going to own it and what the issues are with there. Taking climate change, which Fiona's going to talk about. So these are hydrodynamic models of climate change with different levels of sea level rise added on to look at what that impact will be to our treatment sites and wetlands. And then we're trying to, in the long term, bring a lot of this knowledge into all the ponds and the guidelines that you have here for what people should be doing to design these sites better. So hopefully when we come back, we can say, if we looked at your sites, we can find constant problems that if we redesign them in this way, we can have a better outcome, and that probably leads directly into Fiona's talk.
Thank you, William. Amazing to see the scale at which you're working, and then we've got other people working on the genetic scale. So fascinating blend. Finally, Fiona Johnson. She is a senior lecturer at the University of New South Wales, based in the Water Research Centre in the School of Civil and Environmental Engineering. Fiona has over 15 years' experience in water resources and civil engineering, working as a consultant for government and in academia. Fiona's areas of research interest are in statistical hydrology, particularly with respect to flooding and extreme events, as well as in developing models for climate change assessments of water resources systems. Thank you, Fiona.
Alright, so Will said he'd sort of stepped back from the small scale. I'm going to go even bigger scale. So, unlike Tony Abbott, climate change will cause us lots of problems, and very few of the impacts are positive. We've seen this, from the global body, the Intergovernmental Panel on Climate Change, which over the last 5 assessment reports has continually told us that continued emissions of greenhouse gases will cause warming and changes in all components of the climate system. Now, obviously, that being drilled down into the Vicky, the Victorian Climate Initiative, for Victoria, and as Nick already mentioned, it's fairly clear: expansion of the Hadley Cell, changes in storm tracks. There are going to be changes in Victorian climate that will affect water resources systems in particular, and how these sorts of systems then respond to algae and other impacts, and how that comes together. So if we look at that, specifically, for some sort of shallow lake system, we know we'll have higher evapotranspiration, increased temperatures, changes in humidity, probably drier overall in this part of the world, but increased rainfall intensities when it does rain. So more dry days, but heavier rain when it does rain. And so, particularly at short durations, this has a really big impact in urban environments. It can cause, increased flooding risk. But how that interplays in, over an overall drier catchment is still a big area of research interest. As we've already mentioned, clearly higher water temperatures are a big problem for algal growth. And so, how do all these things start to play together? Do we have changes in wind speed? How's that going to affect, the mixing and the stratification of such systems? These are some of the questions where there's not much in the literature. There's a little bit in terms of the impacts of tropical cyclones on algal growth, but very little in terms of differences in prevailing wind directions and how that might affect the design of these sorts of systems. And so this is really where we're trying to take climate science through to engineering design. So we're not just stopping at, well, these are the changes, but this is how then you change the designs and make them more resilient to climate change. What will happen to catchment inflows? How does this affect the residence time? I mean, again, this is the interplay of this overall catchment wetness. What other things are happening in the catchment? How does this affect the frequency of inflows to any sort of system? What about the nutrients or the wash-off? I mean, we've got people, how are people going to respond in terms of urban areas, in terms of what are they going to do with their gardens? How's that going to affect fertiliser use? There's a whole range of different issues that I think we're yet to get down to the details that affect the design of these sorts of systems. I come to the point in mode as well. you know, from an engineering perspective, and this is where this project is really exciting and bringing in a whole range of experts thinking about this. So most of my work is more in this kind of space, thinking about the changes to evaporation, rainfall intensities and catchment processes, and then how we apply that to different systems. So we've done quite a bit of work thinking about how flood changes, you know, how do we think about floods in the future? How do the catchment processes affect this? So, you know, these are sort of different mechanisms that we think about for catchment flows. So we've got a saturation excess flow versus an infiltration excess flow, and depending on how your catchment works, depends on whether, you know, how the changes to the rainfall itself versus the pre-event rainfall, the antecedent conditions in the catchment. How are our models responding to these sorts of changes? And we've done some work, you know, reviewing this in terms of flood hazard for Australia. But I think the next stage is to take this into a water quality space as well. So thinking about our models and our concepts of catchment processes and how we then design based on that. We've also been doing some work thinking about model structures. So here is a range of, this is WB&M, so a very common flood model used in engineering design. This one's GR4J, which is what the Bureau of Meteorology is using for a lot of their water resources assessments, their sort of you know, seasonal forecasting models and water sort of availability. And on the end is the music hydrologic model structure as well. So these are all really common structures for thinking of, you know, for modelling our catchments. But which ones of these are going to be able to reflect the sorts of changes we see in the catchment? WB&M only looks at a very, very simple loss model. It's not capable of looking at the antecedent conditions in the catchment, how wet it's been before the rainfall occurs. But on the other hand, GR4J is a linear model, so it doesn't really respond to the increases in rainfall intensity in the way that we might expect large catchments to do. So we know that if we get double the rainfall, we actually expect more than double the flow because the catchment is more efficient. The, you know, the losses and all the resistance in the catchment is a smaller proportion. But this sort of model is linear, and so maybe it's not going to work as well under these increased rainfall intensities. So thinking about robust model structures is something that's, you know, really interesting to me and my research group at the moment, and something that I think we can sort of apply to these sorts of projects, thinking about then the downstream design of structures and how consultants and developers are presenting these designs to Melbourne Water. You know, are the methods that they're using the best that we can do? So how do we answer these questions? So a big tool for climate change is a general circulation model or a global climate model, and these are three-dimensional models of the Earth's climate and land surface. They go up into the atmosphere and cover the whole globe. These models are still fairly coarse resolution. They've been improving over time. We've seen huge advances in model resolution. I really love this figure. I wish we had one for Australia, but it works really well, you know, over the US. So the very early generations of the models, you can just see the Rocky Mountains, maybe the Appalachian Mountains if you kind of squint. As the model resolution starts to increase, you can start to see where the Mississippi is coming through and this huge area of southern, you know, US that's going to be susceptible to sea level rise, or, you know, the large areas along here where, you know, lots of low-lying land. So as the resolution of climate models increases, we do see improvements, and this means that they are getting increasing skill in being able to model, particularly precipitation because of the strong orographic effects of topography on rainfall. We're seeing improvement. However, it's still not coarse enough. The best resolution climate models still are around 80 to 100 kilometres, so it's a pretty coarse resolution when you think that the whole of Melbourne CBD is just a single grid cell. So what we think of to deal with this problem is downscaling. And so, you know, when we sort of try to reflect this, you know, you've got a single grid cell, but within that you've got all this heterogeneity in terms of the landscape use, in terms of the way that we've observed it, where we've had rain gauges or other, you know, ways that we're trying to compare our climate models to point data. So there's a lot of scale discrepancies, and downscaling is one way to deal with that. just even with downscaling though, we still have a lot of biases in climate models. So my modelling group has spent quite a lot of time thinking about the ways that we can post-process climate model simulations to make them more useful for engineers. So Jonathan has just submitted her PhD looking at how biases might change over the sorts of cycles of the interdecadal Pacific Oscillation over periods of sort of 30 years. Because these are parts of the climate models that we really haven't examined very well. How well do they represent El Nino? How well do they represent multi-decadal variability that we know is really important for Australia's hydrology and the behaviour of systems. We've also done work looking at some really high resolution downscaling over Sydney. So Jason Evans from the Climate Change Research Centre at UNSW has 2 kilometre runs of a downscaling model over the Sydney area. So this is the sort of resolution that you need to get at to be able to model thunderstorms and convection explicitly in your climate model, rather than having to parameterise it and kind of guess what might happen at the subgrid process. So 2 kilometre runs are incredible. They're really exciting development. But despite that, we still have a lot of biases, and so Jean Wine did quite a bit of work looking at how do we estimate IFDs, so the 1 in 100-year rainfall from these sorts of models. And so we have done a lot of work saying from climate models, how do we translate those outputs to engineering design? And then we've got methods that can take our core scale methods and sort of disaggregate it down to scales that we're interested in as well. So we've looked at a whole range of different methods to make climate models more useful for engineers. We also do some work from the other end of the scale. So climate models only get us so far to understanding the vulnerabilities of our system, and so we've taken what we might call a bottom-up approach, where we look at generating scenarios where we can perturb the climate statistics that we might be interested in a way to understand the thresholds or the places where the resilience of our models, where do our models break, where do systems, their behaviour change. And so in this case, we've taken a weather generator that can generate sequences that are consistent with the historical climate, and then we've made some of the extremes wetter, or we've made it have more dry days, or we've sort of had a combined approach. And this is work that my PhD student Claire has been doing, using as a tool to examine the robustness of those models, the rainfall runoff models that I was talking about before. With that, what she's been able to do is look at, well, given 2 models, how much do their results, how different are their results in these future climates under different conditions. So here we've got more dry days, here we've got wetter wet days. When it rains, the rain is heavier. And we see greater discrepancies between the models as these 2 things come into play, because one of the models didn't really have the capability to think about the dry days. But we get big differences in the peak flow compared to the average flow, because the second model had that linear response versus the non-linear response to the really heavy rain. So, thinking about how do we use these sorts of scenarios to understand at what point does the mixing of the lake systems or the shallow lake systems become critical, or how does this interplay of catchment flows affect the residence time? So generating sequences allows us to explore that uncertainty. So this is a really exciting project for us to be able to think about the catchment, the water bodies and those impacts together. And I think really provide some really cutting edge climate science impact work, because the work that's been done to date for algae and water bodies has been quite broad brush. So I think we can get some really detailed engineering design into these projects and ultimately improve the resilience of our designs to make the management of these systems an easier job for everyone involved. So thank you.
Okay, and our last speaker is Chris Williams. So Chris is General Manager of the Integrated Planning Group at Melbourne Water, bringing together the functions which enable holistic planning of existing and new services to our customers. The group covers integrated water management and resource security, energy, resources recovery, livability and environment, the applied research portfolio, and built and natural assets for enabling core service delivery. So join me in welcoming Chris. That's for the slides.
Cool.
mood. So the order of speakers is to progressively move from the sublime to the ridiculous, and I'm right at the end of the list. So welcome to everybody, and welcome to, I guess, one of the first get-togethers associated with this partnership. It's a particularly important day for us, and I'd also like to welcome Michael Wanmaker, who's the Managing Director of Melbourne Water, and here to see the kick-off of this really important research vehicle. So, cyanobacteria, the algae, get a pretty bad rap at times, but we actually owe them a lot. We wouldn't be here if it wasn't for them and their role in oxygenating the Earth's atmosphere. And so in an evolutionary sense, there's a little bit of algae in all of us. And they're the origin of chloroplasts and all those sort of really fundamental life processes. So they do get a bit of a bad rap, but you've got to take the rough with the smooth, I guess. The potential for cyanobacteria and other nuisance algae to challenge the water industry is, of course, going to grow because of some of the factors we've just heard about. As climate change brings drier and warmer conditions and so forth. And if we look forward to some of the big strategic challenges that the industry is facing, be it climate change, population growth, increased urbanisation, and what these really mean for both water availability but also the health of the water-dependent ecosystems. And so this here is a map of the runoff change under just the medium climate change scenario. So as well as the increased temperature, we can expect reduction in runoff, but also what we do get will be more intense bursts. So overall less runoff, but that intensity of rainfall also has a potential to alter the nutrient transport pattern, so particularly important from an algae perspective. And so in response to rapid urbanisation and climate change, the water industry is of course investing in different types of assets and different types of sources and trying to line those sources up with the appropriate end users. And what that's likely to do is see us spending more time managing shallow water bodies, whether that's at a very small scale with retention of water in the landscape from the broader livability benefits and minimising runoff of nutrients, as well as the changed hydrological regime for local waterways, but also retaining them from a water resources perspective to try and offset use of water from the potable supply system as well. So shallow water bodies, for better or for worse, are probably going to be something we're going to have to do more of and learn how to manage well. And of course, that's where this partnership will be particularly important. So if those assets aren't designed well, and we don't take climate change into account, and we don't design them properly with appropriate pre-treatment or ways or tools or means of managing the risks, then it's likely to drive up costs for our customers and also significantly impact on the beneficial use and enjoyment of that water. So what are we doing? Well, we're not sacrificing any goats to any volcano gods like Tony Abbott would suggest. Instead, we're investing in good science to light the way and really give us some insight into the issues and the opportunities and really guide our efforts so that we invest our customers' money wisely, so that we use it in the way that is most efficient and effective for delivering the things that they, our customers, value. And it's pretty difficult to do that if you don't understand it and you can't test and pilot and play around with and model the sort of changes and interventions that we make. And so that's why good science is particularly important. And so really it's in that context that I formally announced the launch of the Nuisance and Harmful Algae Science Practice Partnership. It's a bit of a mouthful. Surely we can workshop the name a bit or something, I don't know. Which is a collaboration between UNSW's Global Water Institute, the University of Newcastle, the Walter and Eliza Hall Institute of Medical Research and Polytechnic Montreal. And really by supporting collaborations across the different disciplines, be it hydrology and modelling, water engineering, aquatic science, molecular biology and health, we aim to drive the engineering and the science and the knowledge transfer that will help Melbourne Water and others reduce the risks and the costs associated with nuisance and harmful algae. And And I'd particularly like to note that, while the partnership is initially focused on the Northern region, we really do anticipate that the outcomes and the insights and the learnings will be useful across Australia as a whole. And just in closing, I think it's important to note that at Melbourne Water, we believe that a lot can be achieved through collaboration. And Tom Stelkamp, I think he's the former president of Chrysler, as one of his quotes was, "The secret is to gang up on the problem rather than on each other." And to finish where I started in evolutionary territory, Charles Darwin told us that those who learn to collaborate and improvise most effectively are those that have prevailed. And so with that, I'd like to formally launch the partnership, and thank you all for your participation, and thanks for coming today. We really appreciate it. There's been a lot of great stuff that we've learned from the presentation so far. So thank you all, and a round of applause for everybody who's been involved in preparing.
Thank you very much, Chris. And very much appreciate having you all here as an audience. And once again, thank you to our speakers for their preparations and travelling, those who came interstate particularly. Now we do have some eats, and we hope you'll stay and ask those questions you didn't get a time to ask during this forum, and just catch up with each other, use the opportunity to chat and build democracy. So thank you very much.